So first, just do this, just because it's due. These ones are actually kind of fun, but it's just like, I just got so much to do. Alright. So it's this physics thing, finish the lab report, study for statics. Really, I should do statics right now, but I don't even know what to do. You need a workbook. Okay. I guess this one's 4, right? So I don't know. 4. Alright. Open the work PDF. The workbook is designed. Okay. Oh, this is great. In the Baltic Sea, the decline in cod populations has led to a major increase in the abundance of large algae. Even though cod don't eat algae, around the Aleutian Islands, a rapid decline in sea otter populations was followed by the elimination of the economically and biologically important kelp forests and many of their associated species. Even though otters don't eat kelp, the invasion of Flathead Lake in Montana by opossum shrimp resulted in the loss of a bald eagle population, even though shrimp don't eat. What's going on in these communities? These puzzles illustrate there are 2 fairly distinct environments in lakes. The mud on the bottom, called the benthic. I forgot about all these terms. Oh god. The benthic part of the lake, and the water part known as the pelagic. The base of the food chain for both environments is primarily composed of phytoplankton, which are single-celled plant-like organisms that photosynthesize. Many species of zooplankton, which are small, odd-looking animals, and they could easily masquerade as cartoon aliens, made their living directly or indirectly from phytoplankton in the water column. Eating zooplankton are, of course, fish. Fish, however, are not found in every lake, and lakes that contain fish are quite different from those that do not. In this lab, you'll explore why there is such a difference. Using a series of simulated lakes that include 6 different species, there are 2 species of phytoplankton, green algae and cyanobacteria, blue-green algae. Both are pelagic, meaning they primarily float in the water. There are also zooplankton species in these lakes. Finally, there are alewife, a small type of fish, and trout. Is that what Alewife is named after? I wanna feel something, but I don't feel nothing. Trying so hard to get over you. I wanna feel something, but I don't feel nothing. Yo, use so techniques commonly used by limnologists. Bro. To examine lakes. Adding and removing species, examining individuals to figure out what different species eat, and analyzing population growth rates and feeding inefficiencies. Your goal is to describe the changes that take place in a lake community when you add fish and then explain why those changes take place. Bro, let me just simulate a differential equation and move on with my day. This is so boring. Bro, guys, honestly, I'm gonna be honest here. Let's webcam it. I don't get why, I don't really get how this thing works, to be honest. I think it's just that I didn't have it. Anyways, I'm burned out of school. I just want to do projects. Yesterday, I was like, there's two things I really want to do. The first is I was sketching out something that would let me move my camera up and down, like lock it, and then it also has a tilt. And I basically figured out the mechanism. It's just a bunch of bearings, two linear bearings and then some sort of wing nut or something. And then I want to build a scale, just a basic load cell, but a scale, so that I can track the mass of my fermentations over time. But yet we have to do this stuff. Alright, here we go. The top-down control model in SimBio Virtual Labs. The top-down control model reproduces some basic behavior of lakes. If you're interested, here's how the model works, minus a few details that you will figure out in the lab. Growth in lakes is usually limited by nutrients, often phosphorus or nitrogen. To make things simple, the whole lake economy in this model is based on phosphorus and nitrogen. Although you can't see them, the lakes in this lab contain pools of these two nutrients, such that every part of the lake always has the same concentration. Individual phytoplankton grow at a rate that depends on each species' efficiency at absorbing each nutrient at its current concentration, according to the Michaelis-Menten kinetics. This is also not even spelled right. Phytoplankton growth is also affected by the amount of sunlight reaching them through the water column. Okay. The other species need nutrients to grow as well, which they obtain directly or indirectly by eating phytoplankton. Each time an individual zooplankton or fish encounters a food item, it has a certain chance of catching the food. When it eats food, a certain percentage of the food item's carbon and nutrients are taken up, and the rest are excreted back into the general nutrient pool. A small amount of each nutrient is continually added to the lake from outside, and a percentage of what's currently in the lake is always disappearing. Okay. The large panel on the left shows the simulated lake. Okay, so what is this? Okay. Okay, so those are zooplankton. So, these are, okay, cool. Alright. The panel on the right includes bar graphs showing the population size of all species in the lake. The above bar graphs is a species legend which contains a button you can click on to learn more about each species. Okay, here we go. So these are the phytoplankton, cyanobacteria. Alright, so, okay. Alright, so can I run the simulation finally? Click the buttons in the species legend to read the library descriptions. Okay, fine. Which of the two is the better competitor under most conditions? Oh my god, dude. Alright. I think this is Spirogyra, isn't it? As a result, while green algae typically outcompete cyanobacteria, this is not generally the case in lakes that are nitrogen limited, where nitrogen fixing cyanobacteria have a competitive advantage. Okay, so most conditions it's green algae, but if nitrogen, actually, can I just do this? Alright, H3. That's pretty interesting, actually. Okay, what's next? Gotta stay focused. Okay. As herbivores and detritivores, feeding on phytoplankton as well as bacteria and decaying organic matter, Daphnia are well adapted to live in algal blooms, a good source of protein and carbohydrates. Daphnia therefore benefit polluted aquatic environments because they are so common. Daphnia are an important link in food chains of many inland water bodies, serving as a resource for numerous predators such as sticklebacks, minnows, the fry of larger fish, and larval amphibians. Daphnia exhibit both sexual and asexual reproduction, and under optimum conditions, females tend to produce eggs asexually, and such asexual populations can grow rapidly. Under more stressful conditions, males are produced and sexual reproduction becomes more common. As a result, Daphnia are frequently used in studies. Okay, what the fuck? Okay, Bosmina, a filter feeder in the order Cladocera. Bosmina vary in size from females typically outnumber males in the population. Like Daphnia, Bosmina can reproduce via parthenogenesis and asexual form of reproduction, where growth and development of embryos occurs without fertilization by a male. Asexual reproduction is initiated by favorable environmental conditions, although typically smaller than Daphnia and often outcompeted by the larger crustacean. Bosmina are able to successfully compete for food under some conditions because they feed differently. Daphnia are more or less stationary and generalized filter feeder that feeds broadly on all the food encountered that is small enough to eat. Bosmina are active swimmers that can feed selectively. Anyway, so I'm guessing that was the second question. That's it. Anadromous. Okay, are we finally ready to do something? Click step 150. Actually, you know what I'm gonna do? Wait, first off, here, wait, I have an idea. This might save me some. Fuck. There we go. Okay. Bro. Alright, so I'm gonna do this. Oh, wait. Okay. Click step 150 on the control panel and run it for 150 weeks. Wow, interesting. I'm so cynical about this ecology class. It's just boring. If I click save, does it... What? It doesn't save? But if I highlight something... Right? How do I highlight something? Bro, what? Whatever. Alright. What are we doing? We're recording the... Okay, green algae, 94 and 6. $63, $252. 3, 2. What is it? 50. 52. Wait, do I even need to do this? What did we learn? Each graph below depicts population size of two phytoplankton species, P1, P2, and two species of zooplankton herbivores, H1, H2, and a lake. If the patterns you observed in your simulated lake are common to real lakes, which graph pattern would you likely observe? What? Exercise 2, adding alewife. Add 3 alewife to lake by clicking the Add Alewife button. Okay, I get it. Alright, so where's the add? Okay, so we run it for however long. Oh great, I have to do this thing again too. It's like the same. We can just do this. Or wait, I think I can just do this. Okay, fine. It's the same shit. Wait, why can't I move this over? Wait, what? Why can't I make this thing smaller? But this doesn't work now? It makes no sense. Okay, fine. I don't actually give a shit. Can I do this? What the fuck? Dude, what is... Oh my god. This is so slow. Wait, it does have a dims. Oh, wait. But I think it should be dims 2, because I think dims 1, yeah, it's this. Yeah, there you go. Okay, so, that doesn't work. Oh wait, I think you have to do dot minus. What? Okay, cool. Isn't this the same thing? What is it in a green algae? I don't actually know about that. Is that true? 100. Yeah, definitely. Total phytoplankton numbers increased. And then zooplankton numbers decreased. Okay. What are we doing now? For example, you witnessed a trophy cascade when you added alewife because the addition of a secondary consumer changed the abundance. What do they eat though? Wait, they feed on zooplankton, so they're secondary consumers? Yeah, they're secondary consumers. Okay. Limnologists have studied trophic cascades by conducting experiments in lakes. By manipulating variables such as the presence, absence, and relative abundance of each species, they can study feeding habits, competition, and other interactions. This actually you learn to build your own experimental lake in which individual species behave the same way they did in your original lakes. They move, live, and die the same, but you're able to control which species are present in the lake. The next exercise, you'll be introduced to a virtual fish tank where you can conduct additional experiments. In the final exercise, you'll be turned loose to conduct your own experiments. Examine the tool panel. The first 6 buttons should be highlighted. Why does it do that? What the fuck is that? That's so annoying. Okay, whatever that was. That was a bug. Select the microscope by clicking the view organism button. Okay, wait, so I can do this. What? Is that it? Oh, but maybe that's because that's the wrong thing. Let's add this. I don't even see anything. I don't even see anything. Dude, this thing is bugged. There we go. No, wait, so I want to see what this thing eats, right? So I can click on it. Oh my god, dude, this software is so shit. Okay, so wait. Alright, so I need to, like, let it go for a little bit. Okay. I mean, wait, let's just, like, I just want to run it. Okay, so that's cool. So wait, so let's just see then in isolation, what the hell does this thing eat? Oh my god, this is so stupid, dude. It's just like not interesting. Okay, what does this thing eat? Bosmina, Daphnia. It could just be relative abundance though. Basically, this gets outcompeted. What does it eat? It doesn't eat anything. It's a primary producer. So what the hell did this thing? The Bosmina, what do they eat? Green algae. But that's just because, okay, so wait, if I do this though, and this, and let's run it. They just die. They don't even eat it. They can't eat it. Why don't they eat it? But if I did this and this, and I do it, they die too. So they can't. But what if I add a bunch of it? Like a ton of it? Do they still just die? Why is that? But if we add a bunch of this, like even a small amount, they grow fast enough that it's not an issue. So it seems like what's happening is these things kind of just don't grow as fast. I don't know, they grow pretty fast. Something else is going on, I think. Because no matter what, if there's only cyanobacteria, they die. So I don't really know what's going on. Okay, so let's so what do we know? We know that we kind of don't even care about cyanobacteria. So just start out with 0 cyanobacteria, and then just Daphnia, and oh no, you can't undo. That's so great. It's like okay, so clearly Daphnia is the better. So it said, okay, close enough. Add 20 of each, right? Okay. And then we let it run until there's 200. Close enough. Now we add 5 Daphnia. 1, 2, 3, 4, 5. This is really the best use of my time. Seriously. This is the best use of my time. Okay, run it for 150 weeks.
Lava, what are you doing out here? It's sprinkling.
Is it? Alright. Oh. Alright. To test which zooplankton is a better competitor, a good experiment is to add equal numbers I think it's probably this one. But I'm not getting 9 out of 10 on this thing again. Okay, in contrast, you'll see that experimental tanks are particularly useful for short-term experiments on one or a small number of individual organisms at a time. They mimic the control laboratory setups ecologists might use, for example, to study food preference, feeding rate, and other aspects of individual behavior. To mimic the conditions of these short-term experiments, the organisms you add to your tank will neither reproduce nor die. In addition, phytoplankton in the experimental tanks do not move. Remember this as you design your experiments. Exercise 4, experimental tanks. That was 1. Okay, so it ate it. Good. I think it was 2, wasn't it? Let's reset it. 1 green algae, 1 Daphnia, step 1. Oh, it took 1. I should just be able to do this, right? No, it always takes 1. Okay, that's fine. Okay, so the only thing that I really... Bro. Okay, I'm down to do that second one. That second one seems kind of interesting. Bing. Bong. Oh, but they don't grow in this scenario. Okay, whatever. So then how do you even test this in the fucking tank? That's stupid. They don't grow, right? What is this, by the way? Oh, it did grow. Alright, whatever. Okay, actually that's a good question. Let's just add a bunch of this shit. Oh, what? Just add a bunch of this stuff. And let's see, do these things die? Okay. So they can survive on it, right? Do they? When do they die out? This is such a waste of fucking time, dude. Wait, I'm in experimental tanks, but this is experimental lakes. Yeah, this is what I want. This is better. The thing is, they just kind of die on their own, right? So these Bosmina don't even eat the... Fuck, it did this thing again. It's so bugged. So let's just run this thing. Oh, this is the sediment though. Wait. So they produce sediment. So if we put a Bosnia here, if we put a Bosmina here and we step it, and then we microscope it, but it didn't eat anything. But what if we put a Bosmina up here? And we step it. How did it get over there? Okay, so it eats. So Bosmina, I don't think. But does this Bosmina eat the? No, so it's gonna die. Oh shit. God damn it. The fourth dimension. Alright, so let's test that. So we're going to go to the tank and we're going to just overlay them. So we have this and this. And then we're just going to add, like, I don't know, some Daphnia. What did they eat? Green algae, cyanobacteria. Cyanobacteria. It actually seems like they prefer to eat cyanobacteria. Oh wow, I was right. Oh my god, I was right. Alright, let's just quickly read this thing. And then I can be done. As you've seen, the influence of top predators can extend across multiple trophic levels.
Mm, I have no idea how to answer that. I don't think that's right. The graph below presents population sizes in an experimental freshwater stream with two species of algae and two species of aquatic insect larvae that eat the algae. Which of the following are possible explanations for the pattern illustrated in the graph? I don't know. It could probably be all of them.
Actually, this one doesn't make sense. The graphs below illustrate population sizes in an artificial lake. Before and after a top predator, a minnow that eats zooplankton was added. Which explanation below is consistent with the patterns shown in the graphs? This is definitely true that the predator preys preferentially on zooplankton 1, which outcompetes zooplankton 2 in the absence of predators. The predator feeds equally on both zooplankton without preference. Zooplankton 1 outcompetes zooplankton 2 in the absence of predators. That could be. But then what explains the...
What's the best way? Placed in equal numbers. I mean, you don't actually even know any of this stuff, bro. So stupid. Because, like, how do you know that Zooplankton 1 is not just eating Algae 1? Algae 1 outcompetes by a mile. So then, you know, it's like, you have no way of knowing. So, what the fuck is the point of this? A hypothetical grassland system contains 2 species of grass, 2 species of rodent. Okay, so we're given that I mean, because how the hell are you supposed to know that? This literally doesn't make sense. In a system of 4 trophic levels, exhibiting trophic cascades, what would you expect to happen if a species at a 5th trophic level is added to prey on Predator 4? Okay, Predator 4's population will go down. Predator 3's population will go up. Predator 2's population... So it oscillates? Like, what are they even expecting us to say here? Alright, so this goes down, up, down. So it's not this. So this is down. So this could be up. Down, up, down. Wait. Down, up, down, up, down. Up. So stupid. Did I put that one in already? Okay. I think this one's right, because they basically asked us this one. The graph below presents population sizes in an experimental freshwater stream with 2 species of algae and 2 species of aquatic insect that eat the algae. What are the possible explanations? So larva 1 is a superior competitor to larva 2, and it prefers to eat algae too. So that assumes kind of that maybe these should be stable, and that this one is just taking up this. Then the other one, it's like, okay, that's maybe reasonable. Larva 1 is a superior competitor to larva 2, and algae 1 is, that may also make sense. Now, larva 1 and larva 2 are equal as competitors, but larva 1 exclusively eats algae 2, while that one eats both. So you would expect that to not really make sense. So that one doesn't make sense. So I feel like this one's probably right too. The graphs below illustrate population size in an artificial lake before and after a top predator is added. Which explanation below is consistent with the patterns of growth? Okay, so zooplankton 2 thrives, zooplankton 1 dies. So obviously, the most straightforward explanation is that the top predator is eating zooplankton 1. And it looks like in the abs that zooplankton 2 either prefers algae 1 or zooplankton 1 just really prefers algae 2. The predator preys on zooplankton 1, which preferentially feeds on algae 1. No, what? The predator preys preferentially on zooplankton 1. Zooplankton 1 feeds not on algae 1, but on algae 2. So this one's not even right. Preys preferentially on zooplankton 1, which outcompetes. It's probably this. This is a dumb question. Imagine a hypothetical 5-species community with 2 primary producers, 2 herbivores, and 1 predator. What's the best way to determine which primary producer is competitively dominant? Okay, set up 5 plots, each with the entire intact community. Compare average population sizes for the 2 plant species. Set up 5 plots, each with both plants and both herbivores, but no predators. I would probably say this. That's probably the simplest explanation. Based on the graphs in question 6, which again show below, is the most reasonable prediction. Placed in equal numbers in an experimental lake with food. I think it's this. A hypothetical grassland system contains 2 species of grass, 2 species of rodent that eat grass, and 1 snake. When the snake is present, rodent 2 is more abundant than rodent 1. And you can't say this for sure, because even though grass 2 doesn't get eaten as much. But the thing is, the stupid thing is that you assume there's a steady state solution to the system, which I doubt is ever true. We saw in the simulation that it was always oscillatory. So this is just like dumb. The whole thing is based on false premises. When the snake is absent, rodent 1 is more abundant than rodent 2. And total grass abundance is higher than when the snake is present. Okay, so it's not that. When the snake is absent, rodent 2 is more abundant. So it's got to be that one then. But I don't know. Which of the following is the best experimental protocol by which to test the preference of an herbivore for one of 2 plants? Place one herbivore This is what I would do. I wouldn't try to determine preference first. I would say, do they even eat it? So we have H, and we test, the first thing we do is we test This one's dumb. How do you know what the best is? I have no idea. Is this all of them? Okay, secondary consumer. Without alewife, Daphne. Okay, here we go. All right, so they did up, down, up, down, up. The number of producers will increase. Okay.
When the snake is present, rodent 2 is more abundant. And total grass abundance is higher. Yeah, I mean, that makes sense.
Place one herbivore with unlimited access to individuals of both species. Count the number of. I feel like that's fine. Okay, I don't know if I got all these right, but. Did I do it? Okay. Alright, whatever. I love that download. It's like, what? Actually, wait. I want to actually go through.
Let's go through all of these. So, I think the pre-class doesn't have anything, right? Can you view it? Oh yeah, it's just like junk. But the in-class, we just open up. Let's just open up one of these. Does it have the solution? Okay, good. Yeah, so.
And I'm gonna get my graphing calculator because I need to learn how to use my graphing calculator hella quick. Take fast cross products and stuff because I've been using just code to write all my shit. Statics of particles. Alright, so I did this one, right? Introduction. And I got this thing. She's so organized. It's actually really nice. Can I look at one of these PDFs? Oh yeah, it looks good. Okay, good. Alright. It actually has the problems in here, but I don't think they always do, so let's just finish this. Wait, what did I just do? Did I just do this one? Forces and moments. A3. Okay. Force couples. Alright, so force couples, 2D equilibrium, and 3D equilibrium. Okay, so I think what I'm going to do is I'm going to go through all of these, I guess. And then I'll do the practice exam. And I'll probably try to read some of the textbook. Okay, so we did. Okay, so now I'll be able to easily see if I missed any. So did I just download in class A6? Okay, so now we just need in class A6S. All right, good stuff. So now I can close all of this stuff and we can just go through it. Let me get my calculator and time to spam. Oh, yeah, so I'm gonna skip these ones. How do you do it again? Yeah, I'm skipping those. Okay, let me go find my graphing calculator. Alright. So, we can't do like easy vector stuff. Okay. Okay, cool. All right, so let's figure this thing out. So let me get my other notebook. All right. Find the magnitude of P so that the resultant force exerted on the stake is vertical. B, the corresponding magnitude of the resultant. Oh, actually, what I want to do is this. Cool. Okay. Wait, what? This thing goes this way. This thing goes this way. No, that's not right. So How do you find the direction though? Okay, I get it. That's fine. Okay, so 1 of 6 down. So this angle is 90. So then this is 110. So it's 110 plus 40, which is 150. So then R is equal to Alright, so how does she want us to do this? Oh, good. Okay. Yeah. Except I got a different answer. Oh, is that because this is lol. Okay, so what this is actually is this one, and this one. Here, this is 150, so this is minus 30. Okay, so this third one is actually minus 30. Alright, so what we just need to do is we need to change this to minus 30. And we get 200.3. And then for the second one, we get 29.8. Alright. Okay, so that's all good. So now it says determine the resultant. So what we have here is so first we want the norm. Can I take dot products? This is awesome. Okay, so you can actually do maps. You can map the sign function over a list of arguments. So now if I do 50 times the answer, 50 times ants. Let's go. That's amazing. Okay, so you can do basic vector operations. Now the only thing that I really need Where's the arrow? How do I get that arrow? List. Okay, so let's type the list. 3, 4. Then braces are okay. Then press stow. Stow. Where is that? Oh, nice. Okay, so now list L1. Oh, sick. Give me a second. So create a program called Norm. How do I do this? So wait, so if I do A, or wait, no, if I do 5, stow, A, shit. Ooh, that's cool. And then I A. Okay, good. Okay, so I'm starting to get how to use this calculator. All right, so I can store values in there. Okay, so this is what we're going to do. So how do I do this? Program norm. It just says done. So so I need to make it display. So, program, edit, clear, delete. Okay. So how do I do this? How do I do display? Do I just literally write out? Should I go to program? Oh shit! No way! You can write loops! Holy cow, dude. Wait. Dude! Dude, we gotta do this, bro. This is so sick. Please be for... God damn it, dude. Of course it's only for... Windows. I might just go outside and use my computer really quickly. It would be really nice to be able to just do this. Let's see. What is the easiest way? Wait, what? No way? Bro, what is this? Dude, no way. Alright, let's see if this works. Okay, never mind. This doesn't look as good as I thought. I'm gonna go download the... What is this? Because it would be so nice to... So wait, hold on. Let me just see if I take a list and square the list, does it implicitly map the function over the list? Oh, beautiful. Okay, then how do I do sum? So if I took this, I go here and I insert a list operation called sum. So the sum squared. Okay, beautiful. And then all I have to do is wrap this. So if I do second left, oh, dude, I'm getting so good at this calculator. Y'all aren't ready for me, bro. Y'all are not ready for me. Actually, let's delete this. Let's insert parentheses. Insert parentheses. Power 1/2. Actually, this thing is pretty powerful. Okay, so I'm going to be back in a little while when I learn to program this calculator. And this is not cheating, because this is what the calculator people want you to do. Define a norm and a normalized function. The only thing is, I wonder how hard it will be to define a normalized function. But it's really useful to have a normalized function. I wonder how far you can take this. Like, what does the solve button do? No way, dude. There's apps in here? Sigfig calculator? What the hell is that? You can do ANOVA? What the hell? Wait, what is ASM? Is there a way to get help? Okay. Click plus on menu item for catalog help. Oh, sick. Okay, so we go to catalog. We go to ASM, and then we go to plus. Oh, literally, it's assembly program name. That's pretty crazy. Oh, shit. Day of week? That's crazy. It's actually kind of wild. You can't take the cross product. It looks like we would be able to write a cross product program. It seems like you can write... Dude. What? No, there's literally a. Here we go. Reference guide. Converts a string into an expression. I mean, so there's literally a parser in here. So wait, hold on. So that means that there's a string type. How do I make strings? Where's the double quotes? Oh, I see the double quotes. Okay, wait, hold on. We gotta try this thing. We gotta invoke the parser on the TI-84 Plus CE. There's no way we're not invoking the parser. Okay, so what I'm gonna do is I'm gonna call, I'm gonna go catalog, and then can I jump, or how do I get there quick? Okay, I can just hold it down, but that's weak. There's got to be a faster way. There's got to be a faster way than this. What if I just type E? If I go to second or third E? Oh, good. Dude, they actually, like, low-key, this calculator is, like, really good. Okay, so we got expr function. Now we're going to do double quotes 1 plus 1, close double quote. And we're going to call it. Dude. Okay, so it doesn't hold the expression, but it does evaluate it. So, I mean, clearly it parses it and then it evaluates it. Bro, I could spend all day on this. So I think I just got to take a pause. We're just going to define... I think what I want to do is define just a couple of functions. Probably cosd, or just like, or no, not cosd, cs cosine, that basically just maps, takes a single argument, angle, and just applies cos and sine into a list and stores that into some designated variable. Then the second thing I want is cs0 or something like that. That is like a planar version. So it just makes cos x sine x 0. But actually, I don't know how much I want that, because the most important thing is for me to be able to take cross products. So I'll probably want to define a cross product. And what I'll do is I'll just write the code to do the determinant. But you know what's hilarious about this? I'll definitely have a bug in my code, and then I'll just get everything wrong on the exam. So that's like the most likely outcome, is that I write this code wrong and then I just absolutely bomb the exam because I was trying to be smart. But yeah, we'll try to define a cross product that takes two lists, or we I don't know. Let me see here. Is a list of lists, like if I nest a list, can I store that into a matrix? That's what I want to know. Okay, so we're going to make a 2x2 matrix. I kind of doubt this is going to work. We're going to go we're going to go 1, comma 2, close it, comma 3, comma 4, and then we're going to close them. Okay, so that already errored. So you can't... So nested lists are not matrices. So then how do you write a matrix? It looks like there's a function called matrix to list. So can we see that? Oh shit. Okay, wait. So wait, hold on. So the brackets. So let's try this same thing. One. How do I do space? Where's space? Oh, here we go. That didn't work for me. Oh, wait. Wait, matter to list. Oh. Wait, so I think I will be able to, at the very least, but how much time is that going to save me? Matter lists converts columns of a matrix into TI list variables. Oh, and I can store them though. That's like pretty nice. That's pretty sick. So what does list matter do? Because I thought I saw list matter. Okay, wait. So what we want to do is, okay, so here's what we're going to do. We have L1, right? Oh, dude, I'm going to be so freaking goated at this thing. Okay, so what I want to do is matrix math list matter L1 comma L1 comma A. And is A, I mean, I think it's just any, I think it's just any variable, right? I mean, I got a syntax error. No, you know what it is though? But if I do, no, so okay, so there's a special expression that they render out. So you have to go to matrix A, and if we just, can we change the, so this is a 3x3, so presumably that so what we need to do we need to change the shape first, I guess is what I'm wondering. So here, instead of this, I'm going to do an insert. I'm going to insert this, and I'm going to delete this. So, oh shit, wait, so now, second matrix A, bro. Wow. My mind is blown. This thing is pretty powerful. You can definitely do a lot with this. So for cross product will just do, the way it'll work is it's going to call list matter on 111, then L1 and L2, or two lists, and then it'll store that in A, and then what it'll do is it'll just call det of A, and it will store that in B. or something like that, and then it'll just display B. Disp B. Should I end the stream and go stream it over there? Oh, I'm very excited right now. Okay, we're gonna go do it.